So today we are going to know about immunity. Immunity is broadly defined as a capacity of the body to recognize material as a foreign of itself, or to neutralize, eliminate, or metabolize them with or without the injury of its own tissue. Immunobiology is a study of organization and function of immune system. An immune system provides immunity, that is protection against a disease. Jenner, the father of immunology. Edward Jenner in 1749 to 1823, it is the birth and death year of Edward Jenner, is considered to be the father of modern immunobiology. He demonstrated that inoculation of cowpox crust afforded protection to humans against the smallpox. So he was the first one to vaccine for protecting the humans against that smallpoxes. So he observed that a milkmaid who recovered from the cowpox never contacted the disease smallpox, means the person who was with the cows did not have this disease smallpox. He never got that disease of smallpox. So hence the name vaccination from the Latin word vacca for a cow came in the beginning. So the milkmaid and the vaccinated individual were protected from the smallpox virus. Such protection gave them all the immunity to the smallpox. Although Jenner neither knew the actual causative agent of the disease nor the actual mechanism of protection. Now, what is self and non-self? Basics of all the above mentioned protection was the ability of the immune system of the milkmaid and vaccinated individual to distinguish between their own tissue, that is self, and non-self component outside of the tissue, so that is smallpox virus. So an individual induces a physiological response, that is the immune response to a substance that are different from the self component. For example, an immune response is induced against a pathogen, bacteria, virus, fungi or parasite attacking the body of the host. Let's know about different ways by which the body defends itself from pathogen and other harmful substances. There are 4 defense mechanisms of a body. Immunity to defend the body from infection. Metabolic defense to metabolize and detoxify foreign chemical. Stoppage of bleeding, that is hemostasis, to prevent a blood loss. And final, the resistance to stress, mainly through release of an hormone. So immunological defense in most important defense mechanism. It provides protection against various infective agents, for example virus, bacteria, fungi, parasite, and all, also against development of the tumor. So immunological defense serve 3 main functions: defense against microorganism, recognition and destruction of the mutual cell or mutant cell, and removal of a damaged or non-functional cell to maintain the normal state, that is the homeostasis. So the immune system. Now we know that immunity to an infection is one of the most important factor for an survival of an individual. So immunity is mainly provided by a complex network of cell, tissue and soluble factor, and this network is collectively we can call it as immune system. The cell participate in the immune response and organize into discrete lymphoid tissues or organ. So tissue and organ involved in immune system are lymphoid organ. They are divided into 2 groups. First is the central lymphoid organ, that is also known as the primary lymphoid tissue, and the peripheral lymphoid organ, that is the secondary lymphoid tissue. Now let's know some example of the central lymphoid organ. They are the thymus and bone marrow, whereas the peripheral lymphoid organ, they are also known as the secondary lymphoid tissue, example spleen, then mucus associated lymphoid tissue, then peric patches, tonsil, lymphoid nodes, which is associated with the respiratory system, alimentary canal and urogenital. So let's see the diagram of major lymphoid organ and tissue. Here there is secondary lymphoid tissues are present and primary lymphoid organ or primary lymphoid tissues are present. Now, cell of immune system. There are various cells of immune system. We will go one by one. First is the lymphoid cell. It is also known as lymphocyte. So all these are initially derived from the blood cell producing stem cell of the bone marrow. Stem cell means undifferentiated cell which can undergo unlimited division and can give rise to several different cell types. Apart from producing the lymphocyte, bone marrow stem cell are also differentiated to produce RBC, blood platelets and WBCs. So here WBCs are also known as erythrocyte. Blood platelets are also known as thrombocyte, and monocytes are WBCs, and granulocyte are also there. So the macrophage, they are derived from in macrophage. What is it? So lymphocytes are the major cell type responsible for performing immune function. About 10 raised to 12 lymphocytes constitute the mature lymphoid system in the human. So lymphocytes are divided into B cells and T cells. So B lymphocyte and T lymphocyte. So these cells cannot be differentiated, but functionally they are distinct. So cells of the immune system are differentiated on the basis on presence of or absence of a specific cell surface marker. B lymphocyte main function is to initiate antibody mediated immune response and transform into the plasma cell with secrete antibody. Now what is the origin of B cell? B stand for bursa, which studied in a bird showed that the bursa of fabricus, a hindgut lymphoid organ, was the site of antibody production cell. This was the first site where we got the B cell. These cells are therefore termed as B cell because they are derived from the bursa of fabricus. The B cell mature in the bone marrow and then carry by the blood to the peripheral lymphoid organ. In mammals, B cell lineage being in a foetal, that is embryonic liver, and this process began during the 8th week of human pregnancy. The foetal liver continued to be the major site for production of the B cell until well in the secondary trimester, that is the 4th to 6th month, and stem cell then populated the bone marrow, and therefore the B cell are continuously produced in the bone marrow throughout the life. So here we can see a diagram where bone marrow will give the hematopoietic stem cell and burst in the bursa in the bird or mammalian equivalent, and thymus and various blood elements and immunocyte complexes are present. These are formed. So what are the characteristics of B lymphocyte? B cells. B cell display immunoglobulin as an integral protein of the cell membrane, and this surface immunoglobulin antibody act as a receptor for the antigen specific to it. Means it will get attached to that antigen which is specific to that particular antibody. So B cell are responsible for the production of antibody. Activated B cell transfer into the plasma cell. Now B cell differentiated and antibody produce. Now we know that plasma cell produce thousands of antibody molecules per second before they die in a day or so, and some of the B cell progeny do not differentiate into plasma cell but rather become memory cell which produce antibodies in the event of an antigen reappearing again in the future. So T cell. This was a short note on the B cell and their characteristics. Now let's know about the T cell. If we have seen the B cell, on the other hand, lymphocytes leave bone marrow in immature state during the fertile or earlier stage of life, and these are carried by the thymus, mature in the thymus, and then they migrate to the peripheral lymphoid organ. And these cells constitute of secondary major class of lymphocytes, the T lymphocyte, T cell, and T derived from the thymus. So production of T cell is completed earlier in life. But like B cell, they also undergo mitosis in peripheral lymphoid organ, the daughter cell being identical to the original T cell. The main function of T cells is regulate immune response. mediate cell-mediated immune response, induce B cell to produce antibody. T cells are functionally classified into Th, Tc and Ts. That is helper T cell, cytotoxic T cell, suppressor T cell. So helper T cell promote response to B cell resulting in antibody production, active other T cell. Cytotoxic T cell kill virus infection cell and tumor cell, whereas the suppressor T cell, their role is to suppress the helper T cell and may also be B cell to limit the regulatory and activity of the later. So when we see that the T cell mediated two general type of immunological function, the effector and regulator. Structurally, T cells are differentiated on the basis of the presence or an absence of a specific surface molecule, that is the T cell receptor. But both the B and T cell work in cooperation. Antigen antibody. Now let's know an antigen is any foreign molecule that can trigger a specific immune response. Most antigen are either protein or a very large polysaccharide. Another term in immunogen is also used for antigen. However, there is a slightly difference between the two. Immunogen describes a molecule that provokes an immune response, while antigen describes a molecule which reacts with an antibody production. So, paratopes and epitopes. The part of the antibody molecule which make contact with the antigen, it is paratope, and the part of the antigen molecule that makes contact with the paratope is an epitope. Now these are important definition. What is a paratope and what is epitope? Again, antibody, this is an antibody molecule which can make contact with an antigen. It is known as paratope. Whereas here the antigen molecule that make contact with the paratope, it is called epitope. So there may be series of epitope on an antigen. Such epitope cluster, and these cluster are called antigen determinant. Now what are the requirements to become an antigen? The substance should be a foreign in the host. Then molecular weight of the molecule should be 100,000 Dalton or more, and it should possess chemical complexity. Then antibody is a protein molecule produced in animal in response to the antigen. So antibody belong to the category of protein called immunoglobulin, and each antibody molecule is composed of 4 interlinked polypeptide chain. The 2 long chain are heavy chain and 2 short chain are light chain. An antibody has a stem called Fc protein, which is comprise the half of the 2 heavy chain and 2 prongs, the amino acid sequence that bind the antigen. So amino acid sequence is Fc portion and are identical, which is constant for all the antibodies of the same class. In contrast, amino acid sequence of an antigen binding site vary from antibody to antibody in a given class. So let's know the various types of immunoglobulin. There are various classes, 5 major classes of antibodies, namely Ig, IgD, A, E, G, M. These are the various names of the 5 major classes of antibody. They are different in molecular weight and function. IgG is found highly concentrated among 70% of the total immunoglobulin in human. Check out Biomindscape for the various type of immunoglobulin. Antibody are produced by plasma cell, which are differentiated in B cell. So each B cell type produce antibodies which react with a particular epitope of an antigen. Now secreted antibodies travel all over the body through the blood and reaches antigen of the kind thus stimulate the immune response. They combine with an antigen, then direct an attack by the pathogenic cell, which eat up the foreign material and eliminate the antigen or the cell bearing them. Then types and immune responses. Now immune response can be classified into non-specific and specific. So non-specific immune response are those which non-selective protect against the foreign substance. They are not selective against the foreign substance or cell without having to recognize the specific identity. So, phagocytes, that is engulfing of a particular matter by macrophage and extracellular killing by the protein known as complement. So, these are the two non-specific type of immune responses, and specific immune responses are adaptive immune responses. They depend on the immunological recognition of a substance or the cell to be attacked. Specific immune response are again two types, cell mediated and antibody mediated. So cell mediated are the mediated response by the cytotoxic T cells and natural killer cell. These constitute major defense against intercellular virus and cancer cell. And antibody mediated or humoral immune responses, they are mediated by an antibody secreted by a plasma cell which arises from the activated B cell, and they constitute major form of protection against bacteria and virus. So both cell mediated and antibody mediated immune responses are facilitated by helper T cell and inhibited by the suppressor T cell. So cell mediated immune response and humoral immune responses. Let's know the difference. The cell mediated immune response are killing of intercellular organism, destruction of tumor, rejection of graft tissue, and dealing type of hypersensitive reaction after contact with certain antigen. Whereas humoral immune response, antibody specific, combined with antigen which stimulate the production. The combination of antibody with the antigen may result in clumping of a molecule or a particle, and this toxicity may be neutralized. Their uptake and digested by the phagocyte may be facilitated. So combination of an antigen with antibody may cause lysis or a cellular antigen present on the RBC or bacteria, where the lysis is caused in the RBC or in bacteria. So let's know types of immunity. So there are two major types of immunity: natural, which is from birth, and acquired which develop during the lifetime. So natural is also known as innate immunity. A healthy individual is generally immune to potential harmful microorganism. It can fight against any cause of immunity because it have that inner potential to fight against a disease. So this mechanism are termed as innate immunity or natural immunity. So prevent entry of germs, physical barrier, phagocytic cells dealing with the germ which enter, and soluble component, that is the components of this natural immunity. And then physical barrier, it is the first line of defense. Is mean it prevent the entry of pathogen in the body. For example, the skin, epithelial lining of various organ, and body secretion. Now skin is a tough outer layer, is in the form of keratin, and it is mostly important layer of the skin. Sebaceous gland in the skin generate an acidic environment by producing lactic acid which kills many pathogens. Then epithelial lining of various organ, respiratory tract, the alimentary tract, the gut. Then have an exterior epithelial cell layer covered by protecting the mucous lining. In the respiratory tract, cilia covering the external surface of the epithelium cell continually beat upward toward the nasopharynx, and this help in the expel the particle and pathogen. So epithelial cell are constantly renewed and remove the pathogen on the surface. Then body secretions are there, such as sweat and secreting from the eyes also. Then other body fluid contain molecules which are bacterial, that are capable of killing the bacteria. Example, sperminin in seminal fluid, hydrochloric acid in gastric juices. Then if the germs somehow enter the body by invading the physical barrier of the body, so two main defense mechanism come into the play, that is the phagocytosis, where is the bacteriolide effect of the soluble chemical factor, collectively known as complementary system. So phagocytic cells, when a microorganism or inner particles such as colloidal carbon enter the tissue fluid or bloodstream, there are very rapid engulfing and destroying of the phagocytic cells. Such cell may either be circulating in the body fluid or may be fixed in some tissues. This phenomena is called phagocytosis, which means eating by the cell and engulfment and destruction or of the microorganism, which are not important. Now there are two main type of cell, namely microphage and macrophage. So macrophage are certainly in WBC and macrophage are in liver and spleen. Now phagocytic cells, they rapidly engulf the foreign molecule, foreign antigen outcome in contact or with they come in contact, and they contain digestive enzyme to break this engulfed material, and they are imported in the it is an important link between the innate and acquired immune system. Then complementary system, it is a group of protein known as complement, providing another innate immunity mechanism for killing the microbes without prior phagocytosis. Without the phagocytosis, we kill the microbes. So component system is extremely complex system. It consists of 20 proteins. So some of the complementary components are designed letter C followed by a number. So complementary may act as an opson, that is C36. Opson is a type of antibody whose binding of antigen or on the virus or bacterium facilitate their subsequent ingestion by the phagocytic cells. So such antibodies can also cause direct destruction of these microbes by making them memory leaking. Then acquired immunity is immune mediated by lymphocyte and characterized by antigen specific and memory. So acquired immunity may be brought about an individual in two main ways: by infection, so that the antibody are produced against the infective agent, by deliberate artificial immunization, and it is known as actively acquired immunity, and by transferring from an actively immune cell individual through the blood, serum or component, that is the passively acquired immunity. Now important terms is actively acquired immunity and passively acquired immunity. Now let's know what is actively acquired immunity. Due to infection falls into general categories. Some infections such as whooping cough, smallpox, mumps usually induce a lifetime immunity, that is, a patient once recover does not get this disease again. Other diseases such as common cold, influenza, then pneumonia, these are for short period, but they are for few weeks. So these are under the actively acquired immunity. Now passively acquired immunity, it may develop in the form in the way transfer of antibodies, that is IgG, from mother to the fetus across the placenta. Breastfeed children are also receive antibody from the mother's milk. Then pooled human immunoglobulins is also used as a source of antibody in number of cases, including measles infection, infectious hepatitis. Then human immunoglobulin is also given to the patient with a congenital inability to make antibody globulin. Then active immunization vaccine. Active immunization, your vaccination, people have observed in the past that individuals who recover from certain diseases are protected for the lifetime, and this gives rise to the concept of immunization, which was given by Edward Jenner. Introduced the vaccination in 1796 using the cowpox to protect against smallpox. So the objective of vaccine is to introduce the attenuated germ into the body, and the body then generate specific population of memory cell, and these memory cell can rapidly increase in number of renewed contact with the same antigen, and more antibodies can be produced to prevent the protection against infection. Then there are types of vaccine: killed, liver attenuated, then toxoid vaccine. These are types of vaccine. So killed organism as in vaccine, example typhoid, cholera, then whooping cough, rabies or poliomyelitis. Then live attenuated vaccine or weakened organism as in vaccine is BCG. Then measles, polio, these are an example of live attenuated organism as an vaccine. Now attenuation mimics the natural behavior of an organism without causing any disease. So the actively multiplying organism provide a sustained antigen supply. Then toxoid vaccine is an example of tetanus or diphtheria. So toxoid is a chemical or physical modified toxin that is no longer harmful but retains immunogenicity. Then important vaccine is BCG, DPT and MMR. So BCG is Bacillo Calmette Guerin. This is the full form because the name of the scientists for Calmette and Guerin were the scientists who contributed in the development of tuberculosis vaccine, which is BCG. Then DPT is a triple vaccine or antigen for diphtheria or tetanus toxoid and for whooping cough organism. The name of the whooping cough organism is Bordetella pertussis. Then MMR vaccine is attenuated strain of measles and mumps and rubella. So another class of vaccine term as polysaccharide vaccine. They are available comprising vaccine for influenza, meningitis and pneumonia. In this vaccine, the relevant immunogenic portion of this organism are used. Vaccine for future against malaria, leprosy, anthrax and AIDS. So what we learned today? There are various type of defense mechanism in the body. The immunity defend us against infection. The immune system is a complex network of cell, tissue and soluble factors working in close coordination. The thymus and bone marrow are the central and primary lymphoid organ, and lymphocyte which are the major cell performing immune function, and are two main type, B lymphocyte and T lymphocyte. The B cell are transformed into plasma cell which produce antibody. The foreign molecule which trigger an immune response is known as the antigen. An antibody is also known as immunoglobulins. There are 5 types. IgG is found in higher concentration. There are two main types of immune response, specific and unspecific. Then specific immune response can be either cell mediated and or humoral mediated. There are two type of immunity, natural and innate, and vaccine is a type of actively acquired immunity. There are 3 type of vaccine: killed organism, live attenuated and toxoid vaccine. Thanks for watching. Do like, share and subscribe.